Good morning, students. Welcome to Bindu Dhadwal Chemistry Lessons. Our topic for today is proteins of the chapter Biomolecules. Proteins are high molecular mass, complex biomolecules of amino acids present in all living cells. The chief source of protein are milk, cheese, pulses, peanuts, fish, meat, etc. The name protein is derived from the Greek word proteus, meaning primary or of prime importance. So, as proteins are vital chemical substances which are essential for growth and maintenance of life, so they are given this name. Proteins form about 10 to 20% of protoplasm. Important proteins required for the body are enzymes. These act as biocatalysts to catalyze biochemical reactions. Hormones are also proteins. They regulate various body functions. Antibodies are proteins. They protect body against toxic substances and infections. Transport proteins are used to carry different substances in the blood to various tissues of the body. Structural proteins form the structural elements of the cells and tissues, and contractile proteins help in the contraction of muscles and other cells. All proteins contain the elements carbon, hydrogen, nitrogen, oxygen, sulfur, and some elements like phosphorus, iodine, and traces of some metals such as iron, copper, zinc, and manganese. On hydrolysis, protein break down into smaller polymeric forms, peptides, which will on further hydrolysis break down into the smallest units, alpha amino acids. Thus, chemically, proteins are condensation polymers in which the monomeric units are alpha amino acids. Different amino acids, when combined with each other, will release water and join to each other and form peptides, polypeptides, and later on proteins. Amino acids, as the name suggests, are molecules having a carboxylic group and an amino group present at alpha carbon atom, that is at carbon atom which is attached to the carboxylic group. This is alpha carbon atom and amino group is attached here. The general structure or general formula of an amino acid will be a carboxyl group, then the amino group attached to next carbon, and two different groups attached here. There are about 20 naturally occurring alpha amino acids, of which only the first one, glycine, contains a non-chiral carbon atom. Otherwise, all naturally occurring amino acids contain at least one carbon atom, which is chiral. That is, the four groups attached to it are different. Nearly all the naturally occurring amino acids are alpha amino acids containing an amino group on the alpha carbon atom. With the exception of glycine, which has two hydrogen atoms, all other alpha amino acids have a chiral carbon atom and have two optically active isomers. The isomer in which the NH2 is present on the left hand side belongs to the L series, and where NH2 is present on the right hand side belongs to the D series. The L series compounds are so named because they are similar to L-glyceraldehyde. All naturally occurring amino acids belong to L series. So, L-glyceraldehyde has OH on the left hand side. Similarly, L-amino acid will have NH2 on the left hand side attached to chiral carbon atom. For example, this is L-alanine. Nomenclature of amino acids. Although amino acids can be named according to IUPAC system, they are generally known by their common names, and the names are derived from either the name of the source from which that acid is derived, or it's based on the name of some characteristic property. For example, the amino acid glycine is so called because it's sweet in taste, and glycos in Greek means sweet. Similarly, the amino acid tyrosine is so called because it's obtained from cheese, and cheese in Greek means tyros. So based on that, the name is tyrosine. This is glycine, in which there are two hydrogen attached to the carbon, and the formula, and one NH2 and COH is present. So glycine can be abbreviated as GLY, 3 letters of the name are taken, or it can also be abbreviated as capital G. Similarly, this is an alpha amino acid, alanine. Here, NH2 and COH groups are present along with CH3 group. So, depending upon the relative number of amino and carboxylic acids in their molecules, my alpha amino acids can be classified as neutral if they have one amino and one carboxyl group, acidic if more carboxyl group than amino groups, and basic if there are more amino groups than carboxylic acid groups. So glycine, alanine, valine, these are neutral amino acids because they contain only one COH group and only one NH2. These aspartic acid and glutamic acid have more carboxylic acid groups than amino acid groups, so they are acidic. And lysine, arginine and histidine are basic amino acids because they have more NH2 groups than carboxylic acids. Amino acids can also be classified as essential and non-essential amino acids. Non-essential amino acids can be synthesized within our body, so we do not need to take them in our diet. So certain amino acids which can be made by our body and therefore we do not require them in our diet, they are called non-essential amino acids. So out of the 20 amino acids, 10 are non-essential amino acids: glycine, alanine, proline, serine, cysteine, asparagine, glutamine, tyrosine, aspartic acid and glutamic acid are non-essential amino acids. The other 10 amino acids must be supplied in our diet as they cannot be synthesized by our body. These are called essential amino acids. These are valine, leucine, isoleucine, arginine, lysine, threonine, methionine, phenylalanine, tryptophan and histidine. These are required in our diet. The lack of these essential amino acids may result in a disease such as kwashiorkor. Properties and structures of alpha amino acids. Physical properties of alpha amino acids. The amino acids are colorless crystalline solids soluble in water and have high melting points. They behave like salts rather than simple amines or carboxylic acids. This behavior is due to presence of both acidic and amino group in the same molecule. So these amino acids exist as dipolar rather than existing as neutral molecule. They exist as dipolar ion called zwitterion. This is a non-polar molecule where we have one carboxylic acid group and one amino group, but actually the H of the carboxylic group is given by carboxylic group and taken by NH2. As a result, there are two opposite charges lying in the same molecule, one positive, one negative. Such structure is called a zwitterion structure. So it's having two poles, positive and negative, so it's a dipolar structure. So the dipolar structure is also called zwitterion. It's also called internal salt. All alpha amino acids exist as dipolar ionic forms. In alpha amino acids, the acidic character is due to NH3 positive group. That is, this will give proton. And basic character is due to CO negative group. This will accept proton. So basic nature is because of carboxylate ion group. When the solution of amino acid is made, acidic or acid is added to the amino acid, the CO negative will accept proton and get converted to COH. So this site is acting as acceptor of proton and is acting as the basic site. The basic character is due to CO negative group. So CO negative group of the Zwitter ion will accept proton to form a positively charged cation. And when alkali is added, the NH3 positive ammonium group will release the proton and will act as the site for acid. So changes to amino group. So here this will give proton and act as acid, and NH3 positive will be the acidic group. Isoelectric point of amino acids. So amino acids have zwitterine structure, where this is the negative part and this is the positively charged part. So we expect that the aqueous solution would be neutral in nature, but aqueous solutions are slightly acidic in nature because this group is stronger than carboxylate ion group. So when dissolved in water, this molecule, which should be neutral, is actually acidic in nature. This is stronger group. So when we place aqueous solution of amino acid in electric field, its behavior will depend upon the acidity or basicity of the solution. In alkaline solution, this will give proton and form NH2. So the molecule gets negatively charged. So in alkaline solution, the amino acid will behave as a negatively charged ion and will be attracted towards the anode. So it will migrate towards the anode. Whereas in acidic solution, it will take up a proton and will form a positively charged cation and will get attracted by the negatively charged cathode, and so it will get attracted towards the cathode. So that concentration of amino acid which does not migrate either towards the anode or cathode will be called the isoelectric point. So in acidic solution, this behaves as a positive ion. In basic solution, this behaves as a negative ion. But as neutral molecule, at a particular hydrogen ion concentration, it will exist as a neutral molecule and will not migrate to either electrode. And in this form, the structure 2 and the structure 2 and the structure 3 will be in equilibrium form, and there will be no migration, and the pH where there is no migration is called the isoelectric point. So the pH or hydrogen ion concentration of the solution at which a particular amino acid does not migrate under the influence of electric field is called isoelectric point of that amino acid. The isoelectric point depends on other functional groups in the amino acid. The neutral amino acids have isoelectric point between 5.5 to 6.3. For example, glycine has isoelectric point as 6.1. Acidic amino acids have isoelectric point between 3.2 to 3.5, lower pH. For example, aspartic acid, an acidic amino acid, has isoelectric point 3.0. And basic amino acids have between 7.6 to 10.8. Lysine, a basic amino acid, has isoelectric point at pH 9.6. At isoelectric point, amino acids have least solubility in water. So this property is used for separation of different amino acids obtained from hydrolysis of proteins. The test used for detection of amino acids is ninhydrin test. All amino acids on treatment with ninhydrin, that is 2,2-dihydroxyindane 1,3-dione, will give purple coloration, which is used as a test for detection of amino acids. Peptides and proteins. Peptides are compounds obtained by condensation of two or more same or different alpha amino acids. The condensation occurs between amino group of one acid and carboxyl group of another acid with the elimination of water. So OH of carboxylic group of one amino acid will combine with the H of amino group of other amino acid to form water. Water will be given off and these two will join. The point of linkage is called peptide linkage, and the dipeptide so formed and the molecule so formed is called a dipeptide because it's formed by combination of two amino acids. Similarly, if three amino acids combine, it will be called a tripeptide. If four combine, tetrapeptide. Many combine, polypeptides. Now, when the two combining amino acids are glycine and alanine, then this end is called the N-terminal and this end is called the C-terminal. Generally, the N-terminal molecule is written on the left-hand side, C-terminal on the right-hand side. The dipeptide so formed will have the name starting from N-terminal amino acid. So because glycine is placed first, we will give the name glycylalanine. That is, we replace the INE of the first amino acid with YL suffix and we write the terminal amino acid as such. So the two amino acids combine to form this dipeptide. Its name will be glycylalanine. On the other hand, if alanine reacts with glycine, then the name will be alanyl glycine. And in both these dipeptides, there are free functional groups at both ends. The end having NH2 functional group is called N-terminal end, free amino acid at this end, and the end having free carboxylic acid as its end is called the C-terminal end. So a polypeptide will be containing a series of amino acids. The end which has free NH2 group is called N-terminal. The end which has free carboxylic group is called C-terminal. And in between, the CONH link is repeated many times. So this is a polypeptide. The structure is generally written with N-terminal end to the left and C-terminal end to the right. The name of peptide is written starting from this end. So we replace the suffix "-ine" of the amino acid with YL and we write the amino acids one after the other and we end with the last amino acid having the C-terminal end. So for example, this is a peptide formed from glycine, alanine and serine. As serine is having the free carboxylic end or the C-terminal end, the name will be glycylalanyl serine. So this is the N-terminal end, this is the C-terminal end. Similarly, a tripeptide, alanyl glycyl phenylalanine. So alanine is when the R group attached is CH3. Glycine is when the R group is H, and phenylalanine is when R group is CH2C6H5. So the polypeptide will be, this is the N-terminal end, this is the C-terminal end. So name will be alanyl glycyl phenylalanine. alanyl, glycyl, phenyl, alanine. It is abbreviated. The first three letters of the amino acids are named in the polypeptide. So name will be ALA, GLY, phenylalanine has short form PHE. So ALA, GLY, PHE is the name of the tripeptide that is formed by combination between alanine, glycine and phenylalanine. Now, depending upon number of amino acid residues per molecule, the peptides will be called di if two are combining, tri if three are combining, poly if many are combining. The formation of peptide bond can continue until a molecule contains several hundred or thousand amino acids. Relatively shorter are called oligopeptides, larger are called polypeptides or proteins. Generally, molecular mass up to 10,000 are called polypeptides and greater than 10,000 are called proteins. However, distinction between polypeptides and proteins is not very sharp. For example, a polypeptide containing 51 alpha amino acids, insulin is called a protein. Insulin is having 51 alpha amino acids only. Polypeptides are amphoteric because they contain a terminal ammonium and a carboxylate ion. This is the acidic part, this is the basic part. So it's amphoteric. It can react with acid as well as base. They have ionized side chain amino acid residues. So they can behave as acids or bases, and they have an isoelectric point at which it is least soluble, and it has a greatest tendency to aggregate at its isoelectric point. The animal venoms, the toxin in animal venoms are also proteinaceous in nature. They are also polypeptides. And aspartame is a derivative, methyl ester derivative of a dipeptide aspartyl phenylalanine. It is used as a sweetening agent and it has sweetening power 100 times that of sucrose. And the structure is, this is aspartic acid having two carboxylic acid groups combining with a phenylalanine having a CH2C6H5 group along with COOH and NH, and the methyl ester form of this is called aspartame. So this is the formula of aspartame. Then that's all in today's video. If you like the video, please do share and subscribe. Thank you. Have a nice day.